All right. Assalamu alaikum wa rahmatullahi wa barakatuhu. Good morning, boys. I hope everybody is doing well, inshallah. So, today, Age Blue, the topic is about testing the starch in the leaves. So, we will be looking at a simple demonstration, a simple experiment. What are the items required? What is the apparatus needed to check if the leaves are making any starch in it? We know for sure that they are producing the glucose, but is the glucose converted into starch in the leaves? That is the objective for today's lesson. All right. So anybody who is not clear about the objective, you can raise your hand. Okay, so we have one of these links. We're going to start with that link on YouTube.
Do you know that leaves of plants produce glucose?
Small clip for 2 minutes. Let me share that screen with you guys. And are you able to hear the sound? Yes, the sound is on. So we will be conducting a small test where we will be identifying that yes, the leaves are able to convert the glucose into starch. Why do I have two OS? One OS is good enough. All right, so let's start. Let me put myself to mute for 5 minutes and let's continue.
Do you know that leaf plants produce glucose through photosynthesis? The excess glucose is then converted into starch, which is the reserved food material in plants. Let us do a small experiment to test the presence of starch in leaves. We need a leaf, boiling water, alcohol, iodine solution, test tube, spirit lamp. Take a leaf from a plant that has been exposed to sunlight for several hours. Place the leaf in boiling water. This will kill the leaf, disrupt the cell membrane, and soften the cell walls, allowing the iodine to enter the cells and react. Now put the leaf in a test tube and cover it with alcohol.
So he mentioned about two things. He said, number one, we're going to boil it. If we are going to boil it, what's going to happen? The cells will die inside the leaf. All the cells will die. Number two, that the cell walls will get softened up, so that whatever we are going to put in, we will be able to do the chemical reaction with it. We're going to put in the iodine, and the iodine will be able to go into the cells and then react with the starch to change the color. Second thing he mentioned was about the alcohol. Once we put the alcohol, that also helps the cellulose, the cell walls to soften up. And Ghazi has already a question. Yes, Ghazi?
Mister, is that experiment safe to do it at home?
No, this wouldn't be safe to do at home. You have to have an adult supervision if you are going to do this at home.
No, it's Mister. Like with my father. Can we do it at home?
Yes, I'm saying that if you are going to do it at home, you need an adult supervision because it involves the boiling of the water and fire. You might burn yourself. So you have to be careful, and adult supervision is always needed whenever there is fire and boiling of water, right?
Like,
So the things that we need are very simple. You need iodine and alcohol. These are the only two things that you need from the lab. And other than that, please, if you are going to do this at home, I would strongly recommend to have an adult supervision. Is adding the alcohol there? Where did the voice go? Yes, the voice is on. So make sure that the leaf is properly dipped in the alcohol, completely dipped in.
Keep the test tube in the boiling water. The chlorophyll slowly dissolves into the alcohol. Note that alcohol turns green. Removal of chlorophyll will help us observe the color change of iodine clearly. Place the bleached leaf in a petri dish. Pour iodine solution over it. Note the color of the leaf. It changes to dark green. This is because iodine combines with starch present in the leaf and forms a violet-colored complex. This shows that starch is present in leaves. Now cover a leaf with a black paper for a couple of days and do the same test. See if it still has starch.
They should have showed the, what do we call it, the second part where it was covered. That was more important and the guy just simply skipped it. So I'll try to find a video where we can actually see this happening. So let me open up the second video on YouTube where they might be doing this. And we have Ahmed Naseem. So this one is again testing the leaf for starch. Okay, let's see. How does this go?
Hi, today what we're doing is we are testing a leaf for starch. We're testing a variegated leaf, and that is one which has chlorophyll in some bits and not in other bits. I'll just put it there so you can see. In order to do this, we're going to need some hot water, we're going to need a tripod heating equipment, and we're going to need some iodine solution and some ethanol and some safety specs. We'll don the safety specs now. Right. Let's get this water fully boiling. Lighting on safety flame, putting it onto full roaring blue flame. Let's get that heated up. Now what we're going to do is we're going to boil this leaf of this poor draggled plant here, which maybe has seen better days. We're going to boil this in here for about 20 seconds or so. And what that will do is it's going to denature the enzymes, going to kill the leaf, and that will stop photosynthesis. What we're trying to do is see where the leaf is now. So we're going to stop all the living processes going on inside this leaf by denaturing its enzymes. Great, our water is boiling now. I'm going to turn that off here. It's hot enough, I don't need to keep it boiling. I'm just going to get the leaf under the water. There we go. And we're just counting for about 20 seconds. That's all that's needed. It's going to soften the leaf, it's going to soften the cellulose fibers in there as well as denaturing the enzymes. So I think that will do. Now importantly, we've switched off our flame because now what we want to do is submerge our leaf in boiling ethanol. Ethanol, of course, is extremely flammable, and so we don't want any naked flames near it. And we'll boil the ethanol by putting it into our water bath here. So out you come, Mr. Variegated Leaf, pop you in there, and we're going to leave that in there boiling for about 2 minutes. What this is going to do is the chlorophyll doesn't dissolve in water. You know that when you boil broccoli at home, you don't get white broccoli at the end of it because the chlorophyll stays in there. It's not soluble in water. It is soluble in ethanol. And we are now going to remove the chlorophyll. That's not because we're expecting photosynthesis to carry on. It's because we're going to be doing a color test on this at the end of it, and we don't want any green pigments from the chlorophyll in place to interfere with our test. Well, our ethanol is boiling very nicely now. I'm just going to hold this under our camera here. You can see it boiling really, really well. We're going to leave that for 2 minutes and come back to it. Yeah, that is pretty much there. It's done very well. We've done our 2 minutes. We've dissolved the chlorophyll, and I'm going to hold it under the camera here so that you can see that. It's got a little bit hot in the meanwhile, so I'm going to use my tongs. And you can see there, we've dissolved the chlorophyll. It's gone off into the ethanol, and you can already see the leaf is white underneath it. Okay, we're going to pour off the ethanol. We're going to pour that actually straight back into here. And also we're going to put the leaf into there as well. The leaf, well, I'll show you first before I pour it in. It's going to come out all kind of crispy. It's rather peculiar. Out it comes. It's all kind of crispy. It's not floppy at all. Indeed, if I can probably make you hear it. Well, I'm not sure. Here we go. So we're going to drop it into here. That'll soften the leaf up. Because we need it soft, we're going to need to spread it out onto our white tile in order to look for our results. There we go. We'll leave that in there. So, on to our leaf down here. Now, we'll remind ourselves why we're doing this. We're looking to see where the starch is in this leaf. That's because the starch is indicative of photosynthesis having happened. Photosynthesis makes glucose. Glucose is polymerized and stored as starch. So the regions of the leaf where there is starch, that suggests that that's where photosynthesis has been occurring. Okay, let's go. And we're going to put our iodine solution in it, and we're going to drizzle our iodine solution onto it. We'll let it we'll let the stain take for just a few seconds, and that's very beautiful. We can see that really nicely. I'll just zoom into it. Here we go. And you can see here the pattern of the starch. That's the black areas rather than the unstained orange areas. The black areas are where the starch is, and also they correspond exactly to where the chlorophyll was, and therefore this is evidence. It's not proof, but it's evidence that the chlorophyll is required to make glucose, to make starch. The chlorophyll is required for photosynthesis. Thank you.
All right, let's look at these points. We're able to read them properly. He says that we keep the leaf in the boiling water for 30 seconds. Turn off the Bunsen burner because the ethanol is, flammable. Any flame near or around it, even if you are not close to it, it can catch the flame. So that's why we turn off the Bunsen burner, and we boil the ethanol inside the water bath for about 2 minutes. It says 30 seconds, but he actually put it there for 2 minutes. Then we will spread out it on the white tile. It could be a white paper. And then we're gonna put a few drops of the iodine solution. And then we'll see the result. As he mentioned that if it is orange, that means that there are no starch in that region. And if it becomes black, that shows, sorry, that shows that there is starch available. So we have a question. Yes, Qazi.
Mister, a couple last years we learned that plants have two parts, have a male part and a female part. Does does the male part have like just the same as the female part, or it have more or less?
Can you repeat your question again? What is about the male and female part of the plants?
Mister, do Mister the cells inside the male part are slightly different from in the female part, or the same?
The cells are going to be the same, but their functions are different. So that's why they make a female part and that's why they make a male part. The cell will have a little bit of different structure, and their functions is also going to be different. Remember, the cells design the organ. So the cells of the brain are going to be different from the cells of the lungs. So if you are looking at the female and the male parts, they are different. But internally, they will have almost the same things. But the shape of the cell will be different, and the function of the cell will be different.
Okay, Mister, can you show us like what's the structure of a male cell? Because you said it will be different.
I don't think so. I have a picture for it, but I'll have to find it. I'll Google it and then I'll share it with you.
Thank you.
Right, since this is not the topic for today, so I'll skip it. Okay, so let me check if there is a test for starch in leaves where they showed us with two parts, one with the covering and one without the covering. Yes, this one is with the covering, so I'll show you guys with that one. I'll put the link as well.
Photosynthesis is the natural process occurring in green plants that converts solar energy to chemical energy and releases oxygen into the atmosphere. Starch test is conducted to prove that sunlight is necessary for photosynthesis. Initially, destarching of some leaves is done. Some leaves of the plant to be studied are covered with a strip of paper or foil. This is done before dawn to block the sunlight. Any starch present in the plant leaves after destarching is formed under experimental conditions. The plant is then exposed to light for few days. Few uncovered, exposed leaves and leaves that were covered with foil are then detached from the plant. Starch test is then performed on the detached leaves to examine the presence of starch. Detached leaf from the plant is dipped in boiling water for 30 seconds. Boiling the leaf in water destroys the enzymes, thus prevents all the further chemical changes during the experiment. Leaf is then put in ethyl alcohol to dissolve the chlorophyll. Leaf gets decolorized due to the loss of chlorophyll pigment and becomes pale yellow in color. The decolorized leaf is then washed in water and dipped into the dilute iodine solution. Presence of starch can be detected visually by change in the leaf color. Leaf exposed to sunlight turns blue-black or bluish-purple in color, which demonstrates that the photosynthesis was going on in the leaf, therefore producing starch. On the other hand, leaf that was covered does not show any change in the color much evidently, and hence gives brown color with iodine.
So this is an important point. The one which was allowed to do photosynthesis showed us that there was black presence of starch, and the one which was covered in the aluminum foil, it wasn't able to do any photosynthesis. So whatever the starch that leaf had, it started to eat itself because you stop the leaf from doing the photosynthesis. If it's not doing the photosynthesis, it's not making the glucose, it's not making the starch. So where does it have to eat it from? So sunlight is not the starch waste. Sunlight is helping them to do the photosynthesis, so they make glucose. That glucose is stored as starch. Now, if you stop a leaf from doing the photosynthesis, it still needs the food to eat. So what does it do? It starts eating its own starch, so that in 2 to 3 days, the leaf was continuously eating the stored starch in that leaf. So once after 3 days, we put that leaf into the process and put it into the iodine test. It shows negative result. It shows that there is no starch present. Do you guys understand that? So we have Ghazi always asking a question. Nobody asks but Ghazi. That is so sad. Yes, Ghazi.
Mister, okay, Mister. How photosynthesis happen in winter, Mister?
How photosynthesis happen in what?
In winter, Mister. In countries don't have sunlight.
You know, the photosynthesis can happen even if there is moonlight. So
Yes, Mister.
little amount
Moonlight.
of light, little amount of light is good enough to start photosynthesis. Once you covered it with the aluminum foil, you have put the plant, that leaf, into pitch darkness, 100% darkness. So it couldn't do any photosynthesis. But if you have visible light where you can see things, that light is enough for the plant to do photosynthesis.
Yes, Mister. Because some places, Mister, they have inside door farms, Mister.
Inside door, what?
Inside door farms, Mister. Do you not know?
Farms? What's a farms?
Farm, Mister.
Yes, inside farms. Yes, they provide the artificial light. A tube light is good enough for them to do photosynthesis, right? So any feeble light is enough for the plant to do photosynthesis, and then if you can provide them artificially, yes, they would start doing the photosynthesis. So you might have seen that we have long days and small nights, and sometime in some countries there is a very long night and very small day. So in that areas they usually do an artificial farming with the covered areas with the lights provided. So once they have the tube lights, the big floodlights, they start doing the photosynthesis. So let's complete this video and let's see the next 20 seconds. What does it have to say?
Since the covered leaf did not receive the sunlight, no starch is formed in it. So, it is inferred that plant manufactures starch in the presence of sunlight by the process of photosynthesis.
Okay, so let me pause this one here and we'll have a small Kahoot activity. So you guys need to open up your browsers or apps with the Kahoot. I'm going to log in there and we'll have a small 10 points multiple choice activity which is going to help you with your final exam as well, Lab 1 exams. So let's do this. Just give me a minute so I can log in. Meanwhile, anybody has any questions, you can ask a question if you want. Only Ghazi asked the question, nobody else. So Ghazi is having full marks in his participation. And there we go, Ghazi again. The rest of the class is sleeping.
Mister, is not about this lesson, but yesterday I was reviewing science and Mister about diffusion. I know that diffusion happen in gases like oxygen, oxygen and carbon dioxide inside organisms. And they said there's three. What is the third one?
Sorry, can you repeat that question again?
Yesterday I was reviewing
Yeah.
science, Mister.
Yes,
And there's
I understood that part. What was the osmosis and diffusion thing?
they ask about three diffusion happen, three gases that diffuse in organisms. I know oxygen, carbon dioxide. I don't know the third. What is the third?
Three nutrients, glucose. One is carbon dioxide, one is oxygen, one is glucose. They go through diffusion.
Yes.
Blood releases glucose because blood has a high concentration of glucose and the cell has a lower concentration. So that's how it does. So OS has a question. Can we check with OS? Yes, OS.
Mr. The plants it stores starch, right?
Yes, the leaf will store it as starch. Yes.
And what does it do with the starch?
They eat it when they have, enough time. What do you do with the stored
Okay.
food? We have refrigerators. What do we do with the stored food in the refrigerator? We eat when we are hungry, right? So if a plant is not given the sunlight, it would start eating the food stored as a starch. This is stored
Oh,
food.
okay. What if like it is full on food?
So it keeps on storing more food.
How does it create space? Like does it eat
It,
more food?
it grows. It grows larger.
Okay.
It's a living thing. So you can see that they store in their roots, sometimes in their stems, sometimes in their fruits. If it is a root, it would be like a potato or carrots. And if it's a stem, it would be like sugar cane. And if it is a fruit, it could be apple, oranges, or anything like that. So the quiz is ready. Let's start with the quiz. Let me, where did it go? Hold on, boys. Yeah, this one. So let's start with the teach. Okay, so we'll go for this option. The code is gonna appear now. The pin is all right. The pin is in front of you. 6237698. So let me send this out to you guys. It's kahoot.it or the app kahoot.it, and the code is 6237698. So mashallah, OS has already joined. Excellent. Come on, boys, we are left with 10 minutes and we have a 10 minutes quiz. Jawad is here. Mansoor, Basim. Yes, where is Ghazi? Where is the Muhammad Al Habi is here. Good job. Hamza, Yusuf, Ahmed Naseem, Anderson. Come on, boys. Astro Ghazi. Please keep your names, real names, so that I can keep a track of your participation as well. So we'll start at 9:30. Yes, the time is 9:30. Let's begin. This nib cat is out because I'm telling you to keep your names real so I can keep a track of your activities and that you would be marked present with it as well. So let's start with the photosynthesis. I need somebody to read out the questions as well. Who would like to read? Okay, we have the, oh, sorry, here's the mic. Why do we need to boil the leaf to test the
Yeah.
starch? Yes, yeah.
I just wanted to say that my internet connection isn't that good, so I might join like in a few minutes. It's because the Kahoot isn't working for me.
Okay, so let's wait for you. So what do we need to test why do we, where's the question? I can't read the question. Why do we need to boil the leaf to test for the starch? To get rid of the chlorophyll, to kill the leaf, to stop any chemical reactions, and just because we just want to do it.
Yeah.
So, thank you, Thea. And there's another kid raising a hand. And Ghazi has dropped his hand. Okay. So the right answer was to stop any chemical reactions going on. And even if you chose to kill the leaf, both are considered correct. We are stopping the chemical reactions by simply stopping the denaturing the enzymes. So good score, good work. Why is ethanol used? To denature the bacteria, to see if starch is present, to add the color, or to get rid of the chlorophyll. Why the ethanol, when we added ethanol, it became green? So we have 9 answers. All right, the right answer is to get rid of the chlorophyll, the green color. Good job. So who's on top? Okay, Basim is doing excellent, mashallah. Let's go to question 3. Why is iodine solution used? Why am I using iodine? To see if starch is present, to kill the leaf, to remove the chlorophyll, or to denature bacteria. This one is pretty easy with the options because to kill the leaf, we have done that with the boiling of the water. To remove the chlorophyll was done with the ethanol. To denature bacteria, I don't know what is this. To see if starch is present, yes, the starch would be the right answer. So what is your options? And we have 40 seconds left. Yes, 6 of you guys have done it right. And yes, Ghazi, what's the point?
Oh, Mister, just Mister, at first when I entered, so press red, orange, or green. Mister, just my iPad click for itself.
Okay, so it's all right. We will go to the next question. If starch is present, what color will the leaf turn to? Red is dark brown. Yellow says it will turn white. Blue says it will turn purple blue, and green says it turns like blue black. So which one is it gonna be? When the starch is present, what was the color of the leaf? What did it become? Did it become blue black, purple blue, white, or dark brown? Well, last 5 seconds. There's one boy who's still thinking. Okay, good. It will turn blue black, blackish. So those who answer purple blue, maybe I'll consider this right. And white and dark brown, no. So, mashallah, Basim is still on fire. Very good. Okay, video said purple blue. I guess that's what I'm saying is okay. So to test for chlorophyll, what type of leaf should we use? Falcate leaf, non-variegated, and variegated means it doesn't have any veins. Green and variegated. So this is a question is inappropriate for you guys. I don't recommend this to be a correct answer for all of you. So even if you all said it wrong, it will be given +1. So this is out of the quiz. So the right answer is, yes, 10 boys did it. It's the variegated, the one with has a lot of veins and properly. So green, there are a lot of green leaves which are, you know, without veins and with veins. For chlorophyll test, after iodine solution, what color does it turn? It's the same thing. If the chlorophyll, blue, black with chlorophyll and white without chlorophyll, brown without chlorophyll and blue, black with chlorophyll, brown with chlorophyll and white without, and blue, black without chlorophyll and brown with chlorophyll. So for chlorophyll test, after iodine solution, what color does it turn to? Blue, black with chlorophyll, and white without chlorophyll. Brown without chlorophyll and blue, black with chlorophyll. I'm not sure that we have done this test. For chlorophyll test, we didn't do the chlorophyll test, so skip this question. What was I thinking? So 1 second left. This is the answer. Brown without chlorophyll and blue, black with chlorophyll. Okay, so we'll skip this. Next is, if a leaf contains chlorophyll, it is able to photosynthesize and produce starch. True or false? That's a giveaway question. If the leaf contains chlorophyll, will it be able to do photosynthesis and make starch? Is this true or is this false? If somebody does this wrong, then it means he wasn't listening at all. This is the easiest question. Hamza is joining us now. Yes, it is true. The 3 people who did it wrong, please check it again. And Basim is still on top, mashallah. So let's go to, why do you need soda lime for carbon dioxide experiment? We didn't do this, so we can skip this question straight away. And next one is last question. What's the equation for photosynthesis? That is so easy. What are the 2 things which combine to make what? Red, glucose plus oxygen, it produces carbon dioxide and water. Second, yellow, glucose is equal to ethanol plus carbon dioxide. 3, carbon dioxide plus water makes glucose. 4, carbon dioxide plus water makes glucose plus oxygen. So this, if somebody does this wrong, then I don't know how you're going to pass the exam. This is the easiest question. 9 people answered. All right, so 2 people got it right. So how? You have to see it's glucose plus oxygen. So that oxygen is released out of the air. So let's celebrate who has won. Oh, this is the last one. Glucose is stored as, in plants, they are stored as sugars, sucrose, starch, or glucose. Glycogen, sorry. So we have 1 last minute. And that's it. 10 people answered. Let's check the answers. All right, good job. 7 people did it right. So let's see the result. Who's on top? Number 3, Omar. Very good. Excellent. And number 2, Silver, Basim. Oh, Basim came second. And who's on top then? Mr. West. Excellent job, boys. I'll start.